Welcome to Berries in the Interim podcast, where we explore the cutting edge of innovative clinical trial design for the pharmaceutical and medical industries, and so much more. Let's dive in.
All right, welcome everybody back to In the Interim, all things clinical trials, statistics, statistical science. Today we have Frank Harrell. Frank is a professor and the founding chair of the Department of Biostatistics at Vanderbilt University. Helped found the department back in 2003. He is still there. He was a 2014 W.J. Dixon Award for Excellence in Statistical Consulting winner, an award of the American Statistical Association. And what is wonderful, Frank is very much a Bayesian. And for people who listen to this podcast, they know we are Bayesians. We go about the Bayesian way. And Frank is Bayesian. And that'll be part of our discussion today. So Frank, welcome to In the Interim.
Scott, it's a privilege to be here, and it's great to talk with you especially, who's been a force and a pioneer and thought leader and frontlines commander in the Bayesian clinical trial world.
We are fighting this battle, and we're going to talk about that today. So, a good bit of work, and we may even get into things like your modeling of daily ordinal values, which has been really kind of neat. Frank does quite a bit in computational statistics, software, consulting, predictive modeling. But let's talk about a little bit of your career. So you started at Duke and DCRI. So that was right out of graduate school. And your PhD was at UNC, and then you went to Duke. So tell me a little bit about that, and maybe tell me a little bit about Frank the Bayesian. When did that come about?
Yes, so my first real job was at Duke before the Duke Clinical Research Institute existed. And we were in a biometry division and doing mainly cardiovascular research. My whole career has involved cardiovascular research continuously. And it was a great job at a great institution. And I was starting to get frustrated with certain aspects of clinical trials that I just had trouble learning about, namely group sequential testing. And the real general way to deal with that is you have to know about stochastic processes and Brownian bridges of all things to really get down into it. I had a great deal of trouble understanding those things. And I was starting to think there's a cleaner way to approach it. It can't really be this complicated, and it can't really involve so many subjective intentions that have to be factored into calculation of alpha. And so I was really ripe for the picking, and I was influenced by a paper that came out from David Spiegelhalter and his colleagues and JRSS, but something really great happened, which is your dad, Don, came to Duke, and we were both in the same building. So he was in cancer biostat, I was in cardiovascular biostat, and he told me one day, we really ought to meet. I've got something you need to hear. And I went downstairs to his office and had a fantastic meeting, and I came out of Bayesian. At the end of the meeting, I told him I was very swayed by the arguments, and I'm really going to move in a different direction. And to me, that's really a fun thing to change, because some people are just afraid of change. That's one thing I was never afraid of. I got invigorated by change. Even though one of my professors at UNC said that Bayes was evil and should never be used at all. And they didn't teach it. It was pathetic how little coverage. We had 1 week of Bayes in one course. That's just not going to cut it. But Don was very influential to me. And then I started reading more, and I started trying to get my clinical trial colleagues interested in it. And I found something you'll probably identify with, which is the statisticians I work with who were great statisticians at Duke. Chief among them in my group was Kerry Lee, who was, before he retired a few years ago, was like a powerhouse in cardiovascular clinical trial design and execution. And but the statisticians were less likely to change to a different paradigm than the clinicians I work with. They were much more open. But the statisticians always were kind of the, not so much Kerry, but many others that I encountered were more dragging their feet about changing the statistical paradigm. And then the Duke Clinical Research Institute was formed. I was in on the formation of that. I worked in DCRI for several years in cardiovascular clinical trials and loving all that. And the best I could do at that time, I got them interested in bootstrapping as a poor man's Bayes. And so we would take clinical trial results and we would calculate how many bootstrap samples came up with a positive treatment effect as a poor man's posterior probability. And that had some traction there, and not really formal Bayes at all.
Yeah.
But my time at Duke was great. I enjoyed all of it. And then I had a chance to start a new division at University of Virginia.
Yep.
And then I was there. So I was Duke 17 years, and I was at Virginia 7 years, and then I've been at Vanderbilt since 2003.
Yeah. So going back, interestingly, cardiovascular, part of it is the enormous trials. They run very large trials. It's somewhat, the Pocock, Janet Wittes's brilliant statisticians, but still to this day, reasonably frequentist. There's the win ratio test, the Finkelstein-Schoenfeld test, things that almost proportional odds type modeling, things that you've gotten into. I don't know as though a little bit maybe daily ordinal, but the cardiovascular world hasn't changed much in terms of its statistical philosophy since then, or do you think it has?
I think it's prematurely adopted win ratio, and it's leading to a lot of problems. Yeah. So it's interesting they were willing to adopt that in a heartbeat. Yeah. I guess because of advocacy from some great statisticians, and a lot of papers written in cardiovascular journals. But in terms of overall design, it's been very frustrating. And one of the most strange things about cardiology is those massive trials. The typical cardiovascular trial at 6,000 patients to 10,000 patients, they don't need to be that big. But the cardiologists are stuck in their way, so they do time until first event. So if a patient has a heart attack and then they die a month later, the death is totally and utterly ignored. I've been fussing at them about that for a long time, but it so far hasn't taken. But by using time until a binary outcome, they have minimum power, so they're going to have maximum sample size. And also they never do sequential studies. They just grind it out until the end. So their expected sample size is not optimized in any fashion.
Okay, so now you go to Vanderbilt, and Vanderbilt has grown a wonderful biostat department there. Still multiple therapeutic areas, cardiovascular, but in terms of critical care, other areas at Vanderbilt?
Yeah, a lot of us dropped everything we were doing when COVID started to launch this big Active 6 platform trials in repurposed drugs. These were all drugs that none of them should have worked for any reason, and none of them did. And there's another story there that's really worth getting into in the future, but I'll get in trouble for telling that story. So, but we really got into ordinal modeling, ordinal clinical outcomes, longitudinal ordinal outcomes, and Bayesian designs. And we were going to do everything as Bayesian, and then NIH gets involved and they veto a lot of that. So we ended up using Bayesian analyses in one of our key papers to help interpret the results, but we mainly used Bayes for all of the interim monitoring. So it was Bayes interim, using full subjective Bayes, and then it was mainly a defective outcome for the main analyses using things like the Cox model for time until recovery, which recovery is not even a proper outcome in the clinical sense of the word, because we have patients to unrecover, and the unrecovery gets totally ignored. So that's why I've been pushing longitudinal outcomes to the nth degree.
Yeah, yeah. So, and we found ourselves in the same situation where we may design a trial that uses Bayesian machinery, subjective for decisions of continuing or not. And when it gets to the end, it does a traditional analysis. And, a lot of it is the restrictions built around us, whether it's regulators, whether it's NIH, whether it's some other restrictions. And so we're getting closer. Now, your view of that, so you've done the same, where you've, in order to to make progress, you've gone less than what you would like it to be. Do you remember your first trial that maybe was just full Bayes? The whole thing was just full Bayes?
I think it was with the Orbitic group at Imperial College London. So there's this cardiovascular group that is true state-of-the-art thinkers who have no obligation to the past. They embrace change. They embrace innovation. And Roger Al-Lamy and Matthew Shunshin and others in that group, they have adopted longitudinal ordinal to the hilt. They've adopted pure Bayes. And my most thrilling moment, and at some other point I'll tell you my lowest point in dealing with New England Journal of Medicine, my most thrilling moment was dealing with Lancet when we submitted an all Bayesian clinical trial. But we did have one secondary outcome that was analyzed with frequentist methods. And the reviewer for Lancet said, "This is a Bayesian study. Please remove that frequentist analysis from the paper." So then I knew that things were possible.
Yeah.
And since then, Lancet's been sending me some Bayesian studies to review, which I'm really glad to return the favor.
Yeah. And the New England Journal of Medicine does the same now, where we've submitted them where secondaries were going to be frequentists, but the primary is Bayesian, where they want it all Bayesian.
I'm amazed at that. I'm thrilled to hear that.
Let me go back to DCRI. So you must have, cardiovascular DC, you must have overlapped and worked with Rob Califf.
Well, he and I have been collaborating since 1980 when he was a cardiovascular fellow, just having returned from his residency at University of California, San Francisco. So we've been communicating off and on since 1980 and written a slew of papers together. And when he wanted to form the DCRI, my experience in clinical trials had been working as a graduate student at UNC on the most expensive clinical trial ever done, which was the Lipids Research Clinics program, cholesterol reduction trial, and working in the coordinating center. And I thought all the work was boring, and it was very not embracing any new designs. I was kind of bored in that. And I told Rob, don't get into clinical trials, it's boring. So that was because of my exposure. And it was the most ridiculous advice I ever gave anyone, and he totally ignored it. Thank goodness. But I've worked under him at 2 stints at FDA when Vanderbilt puts me on detail, loaning me out to FDA. So I got to work for him twice, and also a thrill to work for Alyssa Lavange at FDA. But working with Rob at FDA was a special thrill, and I never dreamed he would get fired twice and how political things are. But when he was there, it was just a total joy.
Yeah. So he's not a statistician, but do you think he thinks Bayesian and is receptive of Bayesian ideas?
He thinks, I would say, fairly Bayesian, but the thing that distinguishes Rob is that he always believes in division of intellectual labor. And so he said, I'm a cardiologist, I'm going to do my job. You're a statistician, you're going to do your job. And he has just this infinite respect for people of all the different disciplines. And he also has this South Carolina way about him, such that there are people I work with that when they praise me, I feel like dirt. And when Rob told me to go to hell, I would say, by which route do you want me to take?
Yeah.
And so even when he's telling you something you don't want to hear, you feel really good about it.
Yeah.
There's an art to that I could never achieve what he did, but it's just the way he is.
Yeah, a huge respect for him. I listened to him many times when he was FDA commissioner, talk about things. I thought he was fantastic, absolutely fantastic. Okay, so now you are. So, what is this? You're a special advisor for the FDA. What is that? The title?
Yeah, I'm now in my 7th year. Actually in my start of my 8th year of doing this. I did 4 years as a what they call IPA or interagency personnel agreement, where you're paid by Vanderbilt, but FDA pays part of your salary. And I did that for the Office of Biostatistics and Center for Drug Evaluation and Research for 4 years. And then they make you take a 2-year holiday. Can't keep doing that continuously. And then I became, a little over 3 years ago, attached to CEDAR, the director's office, and instead of the Office of Biostatistics, but I work extremely heavily with the Office of Biostatistics and with a lot of rare disease initiatives from the overall Center for Drug Evaluation and Research. So that's as a special biostatistics advisor.
Yeah. So it's interesting. I mean, the FDA, and it's hard to attach any single thing to the FDA. It's a big organization, but over the last 20 years have been much more receptive to Bayesian things, doing Bayesian things. I think on the world stage, in comparison to other regions, are innovating in that space. But relative to Frank Harrell, they're probably not quite where you think Bayesian should be, clinical trials and all of this. But it must be fascinating to work with the FDA that still has great amount of frequentist flavor, to adequate and well-controlled trials. We generally think of this as 5% errors and things like that. How is that going at the FDA? And you don't have to name names or tell stories, but do you feel like you're able to have an impact? There's reception at the FDA?
There's so many good things happening. It's really exciting. And there's a story to be told for another day about the bravery and the stick-to-itiveness of the FDA statisticians in a political environment of great upheaval and uncertainty and fear. A ton of great people are still there, and they're working really, really hard. There's multiple committees in the Office of Biostatistics that I'm taking part in, and these are just thrilling things to watch. There's a group dealing with multiplicity, guidance, supporting documents for how-to for Bayes, and it's just so much going on, and the dedication and the talent level of the FDA statisticians is really something to watch. Now, not all of the statisticians are completely supportive of Bayes. There's some that are motivated by other things that I'm not totally understanding. These are smart people, but they are so much reliant on alpha that it's really hard to shake them. And so I think they'll eventually come around, but I think many of the statisticians are seeing, and we're trying to, I'm hoping someday this is perfectly well defined, the idea that there are Bayesian operating characteristics that don't have anything to do whatsoever with frequentist operating characteristics. And that's a really strong idea to me that could have a lot of impact in the future. And so just getting people to think that there's other design characteristics that you would want to satisfy. And in many cases, it's better to satisfy them than it is to satisfy the classical conditions because they're more concordant with how decisions are actually made and a really key thing that I'm pushing hard at every instance is they don't involve unobservables. So I'm trying, in all of my teaching, I'm trying to educate people about, what sort of things are observable and what sort of things are not. And if you start hanging your hat on unobservables, there's typically problems just waiting to happen.
Yeah. So is this the traditional story that a p-value analyzes the probability of a range of things, many of which didn't actually happen? Where a Bayesian conditions on what happens, makes inferences based on that. Much of it similar stories to this, the likelihood principle. Unbiasedness is even a weird sort of category that involves unobserved things within this. These types of traditional statistical discussions?
It's all of those things, but the central thing is the conditioning on a null hypothesis. Sure. So that leads to a lot of reliance on what might have happened sort of things. And every time you condition on something that it's impossible to know, you're going to be looking at the world through a very strange lens. And if you ask how often that occurs outside of null hypothesis testing, look at how people make decisions in business, in sports betting, in whether to cross a busy street as a pedestrian. I don't think you'll find any other field other than classical statistics where people are trying to evaluate the worthiness of a judge or a decision maker by something that's conditional on something that the decision maker could never know. It's just not done. It's not done in the real world, and it's only done in this small area. So educating people about what alpha actually is the first order of business. And I think it's time for us to take off the gloves because, and I get very nitpicky about this, but when alpha was invented, somebody, I don't know if it's Fisher or who, used the term type 1 error. And it's not an error. It's a trigger. It's an assertion trigger probability. And that has nothing to do with an error. So the rest of the world, when you use the word error, they think of one word comes to mind, which is mistake. So if I want to get a probability of error, it's a probability of making a mistake. And so alpha has nothing to do with that. So when you talk about if somebody makes a mistake, the way that you would calculate their probability of making a mistake is you would not make use of any conditioning about the truth in calculating that probability. So the probability of making a mistake is strictly an unconditional probability, except it's conditional on what information they had when they made the mistake. And so when you condition on what's observable there, you're asking to judge their accuracy in their decision, and the accuracy of the decision can't condition on what they're trying to find out. It's just not the way anybody would look at accuracy. How do you judge who wins, a bet on a horse race or anything you can name? You don't condition on something like that. And so getting people to realize that type 1 alpha is not a probability of making an error is the first step. In other words, you could say that all along, before we even had our Bayesian push in modern times, all along the idea of alpha was misconstrued. It was not something that I would say is a noble cause. It's not a noble goal. It's a convenient goal when you didn't have computers and you could do stuff. You needed to do something with pencil and paper because assuming H naught makes the model so simple that in some cases you can calculate p-values by hand. That it was an expedient thing. And so it was never noble. It was never consistent with how judges need to be judged or decision makers needed to be judged.
Yeah. And now, so you think your work at the FDA, it's having impact. And by the way, I agree with you. The FDA does a phenomenal job. And I think statistics has actually moved quite a long ways from Lissa's time forward in terms of it. We do have a guidance coming out. I know John Scott talked about it at JSM. It's not out, so people can't talk about it, but the expectation is this coming out, highlights for this. This has to be pretty exciting for you.
This is the single most exciting thing. So this is a dream of my career, to be involved in helping with that guidance document. And I have to mention one other important thing. Jack Lee, the great Bayesian statistician at MD Anderson Cancer Center, was involved in this too. So they had two IPAs. Wow. He did an IPA that just ended to the Office of Biostatistics, and I was doing it to the Cedar home office. We were both working on this. So to be able to work with Jack Lee on this, and for the committee to hear from both of us, and we really agreed on everything, and so we sort of reinforced and backed up each other just by accident. That was a thrill. But this being able to have input, even if not all the input is accepted, and we don't know what the final document will look like, to have the opportunity for input and suggesting edits and multiple rounds of things, it was a goal I'd had for many, many years.
Yeah, that is fantastic. That is fantastic. And do we know a sneak peek of timing for this? Maybe that's a hard thing to do.
I think
with
the earliest it'll come out for comment is September. That's this month, isn't it? I forgot. It may be this month. So,
yeah, okay. Yep.
I hope that was the original timetable, and no one has said anything to contradict that. So that's what the hope is. And I can say that the last version I've seen, I'm very, very thrilled with it. It has something for everyone. And of course, I don't speak for FDA in any way, shape, or form for anything I'm saying today. But I think it's a very balanced way to look at it, and it has a lot of operationalizable aspects to it.
That is awesome. I'm excited to get there. Oh, so you are also within the statistics community, one of the more social media active statisticians out there, whether it's Twitter, X, Blue Sky. You have a really nice website with a great deal of material and blogs. And now you're joining a podcast, a new thing. This has been a positive thing in terms of making an impact, I imagine, your social media presence. Do you ever get overly frustrated with social media, I imagine? But this is still something that you do quite a bit.
Yeah, once I quit engaging with people in an emotional way, which is the tendency, everything started working better. It's had a huge impact for me, and I never would have dreamed it. Even just one little example that just reading and subscribing to a good many people on X and Blue Sky, the amount of stuff that I've learned just by watching certain people, and then someone who will post, okay, I have a new course, here's the handouts for it, and here is a new white paper about this problem in Bayesian computation, whatever it is. The amount of things people alert me to is worth it all by itself. But for me personally, what's just as big or bigger is that I got the urge to have a blog, and I put out a blog, and I got like 100 people look at it, and I contacted Andrew Gelman and said, I'd really like to have more exposure. And so he mentioned my blog on his blog, and then I got 1,000 hits that day. Yeah, right. And then I discovered that really it's all about triangulation. So the reason I got into Twitter, which I swore I would never do, I still have never touched Facebook, but that's another issue, but or Instagram. But I got into Twitter to triangulate, and so what X and Blue Sky are really good is pointing people to your more permanent things, your white papers, blog articles, opinions, and course notes, And so I find that social media is just fantastic for pointing people to the blogs, and then also for interacting with people. And people will say, you missed something, and then I'll maybe rewrite part of the blog. And then we created at Vanderbilt a more in-depth discussion that's modeled after the STAN discourse, which is called datamethods.org. And in datamethods.org, we get into extremely long discussions. So there's a very emotional, long discussion now about whether frequentist methods have any role in observational data analysis, and we're sort of concluding it does not.
Okay.
It's not consistent with the sampling, envisioning samples like you need to envision and that frequentist tests really need. But I love these discussion boards, and datamethods.org. We've had discussions that have like 350 replies. You get into like things like odds ratios versus risk differences. It's amazing who comes out to debate that. Yeah. It's incredible. But I found that all of these social media opportunities are really paying off for me because they educate me, and it gives me an opportunity, because at the heart of me, I'm a teacher. So that's what's underneath everything else. So when you're in social media and you think of yourself as a teacher, your classroom just gets bigger.
Yeah, that's awesome. That's awesome. Okay, so we and it's interesting. We within the neighborhood of statisticians, we think Bayesian. We go about designing Bayesian trials. I always look up to you as somebody who is very strongly in Bayesian, thinks that way. There have been times where we may be at odds. Now, compared to what? Sort of thing. Yeah, you sort of thing, but which is all part of this learning and moving forward and doing better. So us that we do a number of these trials and we simulate type 1 error. So we design Bayesian trials, and it might even be the trial is entirely Bayesian. The final analysis is a Bayesian posterior probability. The trial is Bayesian, but we can simulate from a conditioning on a null hypothesis that we've already been through. What you think of that? But traditionally, these are addressed, and we can simulate the probability that trial, if repeated many times, would end up determining a treatment that has no benefit, exactly equal to placebo, say, results in a positive trial. We can simulate other characteristics, really simulate frequentist characteristics of that. Now, we do that quite a bit in presenting to the FDA, presenting to other organizations. We don't do that in every trial. Our REMAP-CAP trial didn't do it in all circumstances, but you would be critical of that act of a Bayesian trial.
Yeah, I think once you give people alpha, all kinds of trouble happens. And I have a blog article that if you haven't seen it, I'm sure you'll find it controversial, because it's about consulting I did with your company many years ago where the simulations caused major harm to the Bayesian procedure because it created a decision rule where the posterior probability was only 0.975. The decision rule was required a 0.986 to preserve alpha. Yeah. And so the study didn't meet its target. And luckily, the advisory committee overruled that and used a secondary endpoint, which is emergency room visits and acute asthma attacks. But it's just an example where just the act of doing that simulation can cause untold damage to the Bayesian decision-making advantages. And so once you give in and you do alpha, you're inviting the consumers of that output to create a hybrid procedure that's very hard to understand, and it's not going to result in optimum decisions, and it's especially going to give the wrong correction for sequential testing. It's going to make sequential testing far, 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 far more conservative than it needs to be, and losing opportunities for really nice futility analyses and whatever else. So this is the only place you and I differ, I think. And so every time we clash, which is not often, It's on a really great topic.
Yeah.
So I've written a lot about how considering alpha, just we shouldn't call it an error. It shouldn't be blessed. It's at odds with Bayes, and I think it's time for Bayesians to quit apologizing. And so my message to the listeners is the reason you choose to be a Bayesian is because you're interested in different kinds of probabilities than classical statistics is interested in. You're not interested in probabilities about data. You're interested in probabilities about unknowns, because the whole thing here, just like when you put a bet on a horse, you don't know if it's going to win or not. You're trying to predict whether it'll win. That sets the odds. And just the idea of dealing with observables and dealing with the probabilities of interest, which is always moving from the known to the unknown. It's always predictive. Predictions are all about predicting what hasn't happened yet or what hasn't been revealed, like a diagnosis of ovarian cancer. It's a prediction problem. And so it's conditional on what has been revealed. And we just have to quit apologizing for that. So if you want to be a Bayesian, I think you should be a Bayesian. If somebody says, I really want to be also guided by alpha, my main response I would hope to give them, without me being on the front lines as you are, is that, you really have just told me you don't want a Bayesian trial. And so you're going to jettison a lot of the advantages of Bayes. You're going to create tremendous complexity. You're going to create an interpretation nightmare. You don't want Bayes. I'm going to advise you to save a lot of money and do something the old way.
Okay. And contrasting that with we may be able to build a better trial doing that. And by the way, the asthmatics trials be really interesting to go back if somebody wants to go look at that. What was fascinating, it was actually more complicated and more on your side of that argument, that the adjustment to 0.986, they enrolled so fast they never actually did the interims.
That's exactly right. Yeah.
So it was alpha
It's a great story.
Yeah, and it kicked the perfect field goal in there. And you're right, the emergency room visits and the other endpoints were highly impactful at the advisory committee meeting and all of that. You gave examples of cardiovascular trials where you're able to build Bayesian trials at the end that did a Cox model in all of that, where you think you can build something better than going back and doing the old way, and you're moving the Bayesian field forward. You're moving it forward with better analyses and better things, and you're trying to satisfy those that want to see an alpha. Now, you're not advertising. You may not be proud of it, that sort of thing. And that's this interesting thing that I always interested in your viewpoints on that, in that part of it, but still feel like you're designing a better trial.
And where you can really unleash the power of Bayes is in two directions, one of which you spend most of your career, which is adaptive trials, and the other one is non-adaptive trials that are highly sequential. Yeah. And so I've been putting most of my effort into, since I'm not any expert on adaptive trials, most of my effort in terms of showing advantages of highly sequential trials with no limits on the number of data looks. And that's where Bayes has a very, very distinct advantage. And then I also put a lot of emphasis on what are the intervals that you want to calculate posterior probabilities of. Mm-hmm. And if you make those intervals bring into the picture the clinical significance, you actually can have futility stopping that is far earlier than people would ever dream. And I think this is in all fields, this is important. It's even an ethical issue. But in rare diseases, my estimate is that of all the studies that end without a positive result, which is the vast majority, they could have had the same conclusion with only 1/3 of the duration of the trial. Yeah. They could have stopped on the average about 1/3 of the way through with a Bayesian sequential analysis that includes a threshold for triviality of treatment effect. Yeah. So I'm spending a lot of effort on on having better clinically relevant decision rules and with Bayes and thresholds and that sort of thing. But as well as really pushing the idea of whenever we have a sample size, we're almost always making it up anyway.
Yeah. Another topic that we have done a good bit of researching in is, and you've recently come out with, I'll call it daily ordinal modeling. It could be time interval weekly, but that a patient has a status on a particular day that's ordinal, one of 5 states, say. 1 is a good state, 5 is a bad state, and then the next day they're an ordinal value, the next day they're an ordinal value. And you're modeling that across the length of the follow-up. Hopefully, eventually they move on and they're out of the hospital. On the other end, they could potentially die in that. But you're modeling that with proportional modeling, which was very relevant in COVID for a number of trials and all of that. We found it very valuable. I'm interested in that. We have found some cases where we actually haven't liked the outcome of it. And in part, it's not the model, but it's the ordinal states themselves. If you end up with somewhat less clinically relevant parts, you break up 1 into 1A, B, C, and D, those alterations can really drive the power of it. So it's still an approach that's driven by a statistical assumption of proportional effect across days and states, which I know you can investigate and all that. I'm surprised that you, as a Bayesian and a subjective, have you spent time trying to attach relative utilities of those states? Where I struggle with ordinal outcomes, yes, this, 3 is worse than 2, but it might be that 4 is much worse than 3 relative to 3 to 2 and 2 to 1, but yet the proportionality doesn't reflect that. Has that bothered you as a Bayesian? And the modeling is fantastic and we use it, but we've run into that issue at times.
Yeah, that particular thing doesn't bother me in the least. And the example you gave where you have some fine grades and those little variations and those might drive the result, that's exactly what you want to happen. You want to get maximum statistical information. That doesn't tell you how to interpret it clinically. So the analogy is when you're doing a blood pressure study, you never round blood pressure to the nearest 5 millimeters of mercury. You use it as an integer, non-rounded variable, and you're going to find changes of 1 millimeter of mercury that are not clinically relevant. But those little bits of information do add enough statistical power to be worth it and to avoid any arbitrariness of lumping categories. And so, but the clinical interpretation comes at the end. Like, what's the reduction in mean blood pressure? What's the posterior mean delta? That's where you put your clinician hat on to look at the clinical relevance. So the way I think about it in particular for the ordinal longitudinal data, which we find the Markov model is the best fitting in terms of the correlation structure most of the time. And it's so easy to program that with an ordinal model. Is that you ultimately would like to have patient utilities, and we find that there's so little research going on to elicit utilities. It's really embarrassing. The clinical trial profession has dropped the ball, except in certain cancer areas where you're trading off toxicities and stuff like that. But in general, I find that utilities are all important and almost never elicited. So I think of the ordinal variable as an approximation to what you would need to be doing. And the question is, what about what you mentioned with these little differences? Well, when you're asking which treatment is better, you don't want to combine any categories. You want to do like the Wilcoxon sort of idea, concordance probability or so-called probability index, which treatment is better than the other. And if you had non-proportional odds, the proportional odds model still tells you the right answer about which treatment is better. So then the question is it enough better to be worth it? And so the only time where the spacings of the categories are relevant or definitely relevant is if you want to calculate a mean or an expected utility. If you want to calculate a median or the probability of having an outcome greater than 10, or you want to calculate a concordance probability like a Wilcoxon sort of thing, the spacings are irrelevant. But if you want to calculate a mean to bring in something on your outcome scale that might be clinically relevant, the spacings come back in to haunt you. And if you wanted to calculate the treatment that has the best expected utility, you can't analyze utilities with an ordinary linear model. They have a distribution that doesn't behave correctly for that. So you have to model utilities with an ordinal model to handle bimodal distributions, floor and ceiling effects. But once you get your ordinal model, now you have ordered categories and you assign the utilities to those, and then your ordinal model will give you the expected utilities, and your Bayesian result would say, what's the probability that the expected utility for treatment B is more than 0.5 units better than the expected utility for treatment A? So the utilities come back in at the scoring point, just as if you were calculating a mean, which needs more than ordinal to carry it off. So I intertwine all these things, and I really agree with your viewpoint about utilities. And I think that until we get the utilities, the ordinal analysis is the approximation that is the best we can do at the moment.
Okay, wonderful, wonderful. Agree with that. All right, Frank, one thing about being in the interim is it doesn't last very long. We're very quick doing interim analyses. So I very much appreciate you joining us here in the interim. Is fabulous. Love to have you back on, maybe after the guidance comes out, and talk about the FDA Bayesian guidance and your thoughts on that.
I would love it.
Fantastic. Well, thank you very much, Frank. Appreciate you joining us.
Scott, thank you for having me, and thanks for the stimulating conversation that you generated.
I enjoyed it. Thank you.
All right. Take care.